In this section, we will discuss about measuring effects of the vaccine in pediatrics, and we will take an example of RTS S age de-escalation studies and challenges of the pediatric vaccine trials in low and middle income countries. I am Nahia Salim. Development pathway. There is ethical tension between favoring the speed to develop a vaccine and being conservative for the sake of the safety of children. So traditionally, vaccines have been developed and first evaluated in industrialized countries with eventual application in developing countries. Development of preventive interventions against endemic diseases in developing countries or conditions with high disease burden, that is public health needs. So this approach provides actually a better knowledge of risk and benefits of the intervention in endemic population, and therefore making informed decision about the acceptable risk and benefit ratio of the vaccine candidate tested. The following factors can influence the development pathway. The health risk between industrialized versus the low-middle income countries. For example, pneumonia and diarrhea affects children living in developing countries and hence less weight in industrialized countries. Additionally, assessment of a causal relationship between a vaccine and observed side effect may be difficult due to naturally occurring pathological manifestations in endemic areas. In countries such as malaria, it may be advisable to conduct parallel trials. So that means parallel trials within the developing countries and the industrialized countries. Traditionally, phase 1 and 2 have been conducted in industrialized countries for assessment of safety in the population who are at low or no risk of the targeted disease, and mostly in adults, as children cannot give a proper informed consent, and also to avoid potential risk of the vaccine before safety evaluation. Let's look at the steps for a malaria vaccine development. There's a preclinical testing, phase 1, 2, 3, and 4. If you look quickly at these phases of development, it takes a minimum of 20 years for a vaccine to be developed to a phase 4. And up to now, it is the RTS S is the only advanced malaria vaccine at phase 3, currently at phase 3, moving to phase 4. In vaccine trials, what do we mean by age de-escalation? Age de-escalation means conducting a phase 1 and 2 trials first among the adults, and then in older children, and finally in the targeted age group, in order to avoid undue risk to the younger child participants. So factors which be considered during this age de-escalation includes the epidemiology of the disease for which the vaccine is prepared, the risk and benefits of the vaccine for each age group, and the safety profile of the vaccine. Disease such as malaria provide partial immunity, and the adults who have repeatedly exposed would have higher levels of immunity compared to younger infants with no prior exposure. So different safety profiles are expected in different age groups, and gradual age de-escalation is desirable. The scientific justification or ethical considerations. For example, a prior immunity in adults would be risky because of the anaphylaxis or hyperimmune reaction because of the previous exposures, or if the vaccine has been extensively tested or used in children in the industrialized countries, then it makes sense to test it first in children in the developing countries. The products which have undergone phase 1 in adults in industrialized countries should be tested in adults in developing countries before they are undertaken in children. Why? Because there may be different safety profiles in different ethnic groups or levels of prior exposures to infection. So let's consider an example of a GSK, again, RTS S malaria vaccine candidate. We see here in 1984, the GSK initiated a collaboration, that is a preclinical study began, and in 1987, actually the RTS formulation was created by combining the malaria circumsporozoid protein together with the hepatitis B antigen. In 1992, the first clinical test in human beings among the adults in the US was done, and followed by a proof of concept among the US adults. Later on, first trial in African began in Gambia, and a proof of concept among the African adults in Gambia was established. And in 2001, a research partnership was initiated between GSK and MVI Path. And 2004, there was a proof of concept among children in Mozambique. At the same time, in 2007, another proof of concept or safety and efficacy was established in African children in Kenya and Tanzania. And in 2009, the phase 3 MAL55 study was initiated, which continued up to 2014, end of 2014, which marked the end of the phase 3 MAL55 study. Actually, it was a controlled double-blind trial with 3 arms. The first arm received 3 doses of a primary course of the malaria vaccine, followed by a booster dose 18 months post dose 3, while the other arm had 3 primary doses of a malaria vaccine, followed by no booster dose. So this makes us able to analyze the role of a booster dose post-primary immunization, and the control group received no malaria vaccine. And actually, the younger infants received main C as a control vaccine, and the older children received anti-rabies vaccine as a control vaccine. Steps have to be taken to establish the following when developing a vaccine or testing a vaccine. You should establish its safety, should establish the adjuvant to be used, the dose, the schedule of the vaccine administration, the expanded program of immunization co-administration schedule, and also the immunogenicity. You should determine the correlates of the immune protection. Shown that, for example, with the RTS S malaria vaccine, the infants have shown lower efficacy compared to the older children aged 5 to 17 months. And this could be explained by maybe the co-administration of the RTS with the routine epi vaccine. So maybe there was interaction between RTS and the routine epi vaccine. and also an absence of priming with hepatitis B vaccine or with a P-falciparum infection, or there was an inhibitory effect from the maternally derived anti-circumsporozoid antibodies, or this was because of the infant's immature immune system, the imbalance between the B and T cells development or the whole issue of maturation of the immune system. Secondly, it has been shown that there was effect of transmission intensity as a potential explanation of lower vaccine efficacy. With that, it has been shown that the vaccine had higher impact among the areas with high transmission intensity as compared to low and moderate transmission intensity. And therefore, this may have contributed to differences in results observed within different sites within Africa. Could also again be explained by the waning vaccine efficacy or the differential acquisition of the natural immunity, heterogeneity of exposure because of the different transmissions intensity between sites, and also heterogeneity of the vaccine effect at the level of the individual. Would also contribute to the lower efficacy observed among young infants as compared to the older children. And this may be contributed by the maternally acquired immunity or the fetal hemoglobin levels and the lower exposure of the infection and other factors associated among the young infants. So the take-home message is the vaccine development requires time, and the efficacy of the vaccine can vary with age and ethnicity, the epidemiology and intensity of the disease, interaction with other interventions, the adjuvant used can vary with time, and also underlying immunity and medical conditions. Thank you.